How are you, my dear students? Today, our new topics is the heredity in organisms and evolution. Here's I just select about the DNA. Here's the DNA. I just explain about the DNA replication and the DNA test. Listen, my dear students, these two topics are very important topics for your final exam. Basically, in this final exam, I just explain about the DNA, replication of DNA, and here's the DNA test. What's the meaning of replication of DNA? Please everyone see the books. Here's I just explain about the replication of DNA. Through the process, a new DNA molecule is synthesized from an existing molecule of DNA. DNA replicates in a half conservative way. Have you any idea about what is the meaning of half conservative way? Yes, the half conservative way is first structure is the mother DNA. This structure is basically the mother DNA, and this structure is basically the daughter DNA, and this explained is the semi-conservative way. Here's the semi-conservative way. Through this process is the double-stranded DNA becomes single-stranded by breaking hydrogen bond. Here's the hydrogen bond. They are just combined basically adenine and thymine and guanine and cytosine. Here's the hydrogen bond. Basically there are two hydrogen bonds and the cytosine and guanine. This structure is the basically three hydrogen bond. When they are when they are breaking, then the one strand of DNA, then it goes to the one strand of DNA. Basically, they are the two strand of DNA. One hydrogen bond is the breaking. This structure is the one strand of DNA. Okay. Then the floating nucleotide in a cell creates new complementary strand by combining A, adenine with T, thymine, and T with A, and the thymine with adenine. Here's the C and G. Basically, C with G is the cytosine with guanine, and the guanine with cytosine. Here's I just see this. Basically, this another structure is the create. This structure is first adenine and the thymine. Guanine is the cytosine and the thymine is adenine. And the another single strand bond, they are also selected the complementary bond. Here's the complementary bond. This structure is thymine and is the adenine, cytosine, and the another is the guanine. Here's then the floating nucleotide in a cell creates new complementary strands by combining A and T, adenine and with thymine, and the G with C, guanine and cytosine. Thus, one of the old strands remains and combines with a new strand to make a complete DNA. This structure is the old DNA and another structure is the new stranded of DNA. At the end, a new strand combining with another old strands forms the structure of a molecule of DNA. As the new DNA emerges, it has a new and old strand. This principle is known as the semi-conservative method. In 1956, famous scientists Watson and Crick first successfully proposed the replication process of DNA. Dear students, I just explained about the replication of DNA. I just explained here's the mother DNA and another is the daughter DNA, and they combined with the new and the old strands of DNA. Our next topic is the DNA test. In the present world, basically, DNA test is the most important and also the famous test. Here's the DNA test. I just explained about the DNA test. Throughout the present century, DNA technology and the significance of it in the field of medical science, agriculture, fish and animal resources, and in the industry of medicine have introduced a new chapter on creation. In Bangladesh, along with eyewitness dependent judiciary system, DNA has opened a new window of ensuring due through justice. A scientific and practical method of DNA test is called the DNA fingerprinting. Basically, DNA test, the another name is the DNA fingerprinting. The name DNA typing or DNA testing is also used. To perform the DNA test, the first requirement is an organic specimen. Basically, in this organic specimen, I just read first structure is the bone, another is the teeth, another is the blood, bone, teeth structure. These also the specimens. Here's the specimens. Basically, the first requirement is an organic specimen, both bone, teeth, hair, saliva, semen of a person can serve as valuable organic specimens. The profile from the place of occurrence of crime or the profile from the specimen of the victim of a crime is compared with the profile obtained from the blood or organic specimen of a in this method. DNA is isolated from the sample by a chemical process and then DNA are cut into pieces. Here's the basically this structure of DNA I just take is from the victims and also the suspected. Here's the first basically DNA. I just cut this 3 or 5 or 4 structure. Again I also repeat in this the method. Here's the profile from the place of occurrence of crime or the profile from the specimen of the victim. Here's the crime and another is the victim of the crime is compared with the profile obtained from the blood or organic specimen of a. In this method, DNA is isolated from the sample by a chemical process and then DNA are cut into pieces by one or more restricted enzymes. You know what is the restricted enzymes? Restricted enzymes, the another name is the biological knife. Then through a special method, basically electrophoresis by agarose or polyacrylamide gel, the pieces of DNA are sorted according to the length of different bands. Here's again I also repeat. Basically, through a special method, electrophoresis by agarose or polyacrylamide gel, the pieces of DNA are sorted according to the length of different bands. Then radioactive isotope DNA probe is hybridized, keeping it on a special nitrocellulose paper and then putting it on an X-ray film or to determine different visible bands by autoradiograph method. Please everyone, again I also repeat, basically it's very important line for your these topics. Again I also repeat, basically through a special method, electrophoresis by agarose or polyacrylamide gel, the pieces of DNA are sorted according to the length of different bands. Then radioactive isotope DNA probe is hybridized, keeping it on a special nitrocellulose paper and then putting it on an X-ray film to determine different visible bands by autoradiograph method. After that, the sample is compared with DNA band or the DNA map of the victim or suspected person. Here the suspected person 1, 2, 3 or 4, but victim is the one. Their method is termed as fingerprint method. Currently, by using polymerase chain reaction (PCR), you know the machine of PCR. In the coronavirus, we also the test about this the machine PCR. PCR meaning is the PCR polymerase chain reaction method. DNA test is done with even a small sample for identification without any error. Dear students, I just explained about the main topics is the replication of DNA and also the DNA test. Here's the DNA test. The another name is the DNA fingerprinting or the DNA map. Dear students, it's very important topics for your exam. Please everyone prepare it again. Thank you. Again, I also repeat this structure. In this method, basically this structure, we also know that DNA test, everyone know that there is the basically phosphate and also the nitrogen base and also the uh, phosphate and also the 5 carbon based ribose. And this structure we also have the replication of DNA. And the another topics is the basically we also take at the DNA sample or the 5 sample to test the DNA test. Dear students, please everyone, these topics is the very important topics for your exam. Again I also repeat this the line basically DNA replication of DNA and the DNA test. Please everyone draw this the picture and also the take it the properly. Thank you.